This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hey everyone, something a bit different this month. We've got a recording that we did last month with Jesse Noar, host of the Microbe World podcast Bacteriophiles, and we're reposting that here now. So we hope you enjoy it. We've got a new episode of Audiommunity with just Matt, Kate, and I coming up soon. But for now, here's Bacteriophiles.
So we talk a lot about how there are good bacteria. And so that's getting more well-known, and I think in the public, maybe thanks to me, partially.
We've had some episodes on it, too.
Yeah, definitely.
Yep.
But it's often harder to see how viruses can be good because they basically depend on healthy cells, or they depend on taking over cells and hijacking their resources. But in this paper, we're talking about oncolytic viruses, which are used to kill cancer cells specifically and leave the healthy cells alone.
So had anyone seen, by the way, I think there was a 60 Minutes or something on the poliovirus trials that are going on right now?
I think I got like 20 emails after that came out.
I'm sure you did. Yeah.
I think Matt did a mini-sode on that on our podcast as well at some point.
Pretty behind.
Yeah, no, I think so. So, yeah, basically it's just this group that has started using poliovirus to target cancer growths and seems pretty effective. It turns out that polio is pretty good at selectively targeting cancer cells. And so if you make slight modifications so that the polio obviously doesn't go systemic and kill you as a person, you can actually do a pretty good job of targeting cancers.
Nice. There's polio. Yeah. There was that measles vaccine modification.
Yeah.
Just for the brain tumors, I think.
Mm-hmm. Yeah, there's some really good data on a modified herpes virus where it's unable to replicate in a normal functional cell. But this herpes, like when it's injected into the brain, it specifically targets glioblastomas. And glioblastomas are these brain tumors that have a very poor prognosis. So most people die after a couple months of being diagnosed. And it actually extended these people's lives by months and months and months. It was, I think, one of the most successful glioblastoma trials ever. But that's just like a modified herpes vaccine, which normally you think about herpes getting some sores on your mouth, but instead they injected it into the brains of people.
Why not get brain sores?
Yeah.
Yeah.
I actually, this isn't oncolytic, but there's also viruses that are just in like the helpful virus category. There's viruses that are used as vectors also for cancer treatments, but they use them to express protein. So they no longer replicate fully, but you can use them as a vector to express a protein that can target the cells. Like either you can give them new function to target cancer cells and kill them, or you can use them as like some sort of marker. So there's like a lot of different things are going on with viruses.
Did I just get disconnected, or was that Kate?
I think it was Kate. Yeah.
What did I do?
Kate,
you disconnected for a second.
Cut off.
We just lost you.
Oh, okay.
But you were making great points.
Mm-hmm.
Do I need to repeat them?
No, I think we got it.
Okay.
Viruses are cool. Should we say what oncolytic means?
Yeah.
So
yes.
Onco is the prefix for all things cancer. Cancer doctors or oncologists, etc. And then lytic is just blowing up. Lysis is basically breaking apart or exploding. That's the way I like to think of it. So oncolytic is exploding cancer.
Okay. I mean,
explosion seems a bit
might
aggressive
be a little bit
for the
more
word
dramatic.
Yeah.
No, I picture little
scale.
explosions, little dynamites going off inside the cells.
Like lysing the cells. I like to think of it as like a balloon slowly losing its air.
Huh.
It could burst.
Yeah, I definitely have burst in my brain.
Huh.
Okay.
I suppose maybe you have your depiction is maybe a little more accurate, but I always think about
sticking a needle in a balloon and having it explode, like a water balloon.
Yeah.
Needle
depends.
That would, I mean, not to go fully immunology here, but that would be a bit of a problem if the cells were just exploding all of their contents everywhere and it was not actually a programmed or controlled destruction of the cellular components.
Although it would serve as a really good damp, though, right?
Exactly. Oh, yeah.
The explosive power of a dying cell activates dendritic cells. I like that.
But maybe not in your brain.
Now I'm just imagining somebody's, like a tumor in somebody's brain just like popping out of their skull.
Okay.
We've gone too far, I think.
Got too far.
Sorry, Jesse, we're hijacking your podcast.
This is great.
Thing.
Okay, so in this particular study, it was looking at cancers of I think it was the immune system itself, like leukemia and multiple myeloma.
Mm-hmm.
Yeah.
So those are hematopoietic cells that are cancerous, I believe.
Yeah, so the multiple myeloma is actually a B cell. It's a plasma cell. So these are cells that are usually going to be producing antibodies. So if you get a vaccine or something along those lines, the antibody responses that we look at, almost all of them come from these plasma cells. So you can imagine that if a plasma cell is growing and dividing and producing antibodies, and then all of a sudden it just keeps dividing and keeps dividing and keeps dividing, and then you get a buildup of these things in the blood, and a lot of your lymphoid tissues as well. That's what we're sort of talking about there.
Yeah, and as I understand, they're usually treated with radiation and then a transplant of the bone marrow, I think, from a donor.
Yeah, and so I think we've discussed this maybe before in our podcast, but one of the in all of these leukemias, basically, you've got an immune system that's growing out of control. And so you mentioned that these things are all hematopoietic in origin, and the beauty of that is that an entire immune system can be generated from a single hematopoietic stem cell, and studies have really nicely shown that all it takes is one cell. And so if you've got one of these cancers of the immune system, what you can do is you can basically just knock out someone's entire immune system. So the radiation is designed to essentially kill off the cancer patient's immune system entirely. So you've got radiation and chemo that are designed to do that. Mm-hmm. And then, obviously there's some severe secondary effects of that. People die of infection all the time during this procedure. But if you succeed in killing off enough of the cancer patient's hematopoietic system, you can basically just replace that with someone else's immune system. So you get a couple of those stem cells from someone else into that system, and because the patient is severely immunocompromised, they basically can't reject that immune system, and so the new immune system just kind of grows out and becomes the person's new immune system. It's kind of neat.
Yeah.
And I should also say that for some of these things, like if the etiology of the cancer is really well known, like if it is a B-cell tumor, these days we have some really cool monoclonal antibody therapies that can target specific subsets of cells. So there's like I think it's like a CD20 or something like that, that targets B cells and causes them to basically undergo something called antibody-dependent cellular cytotoxicity, where they get attacked by NK cells and T cells and exploded. Not actually exploded.
Yeah.
They commit suicide. But that can be a slightly less traumatic way than zapping your entire bone marrow.
Yeah.
But
does it still require the transplant after that?
I'm not sure, to be honest. I think if it works, you don't need additional therapy. But I wonder if for some of these things, like the cells de-differentiate and lose some of these surface markers, so that those aren't as effective, maybe.
Yeah, and I would say that so we're talking about pretty much terminally differentiated tumors. As far as hematopoietic tumors are concerned, there are also undifferentiated, and not tumors, cancers, I suppose. So there are some systems where you can imagine you're constantly turning over neutrophils in your body. Neutrophils have a half-life of about a week to 2 weeks or so in the blood. So they don't last very long, so you're constantly making new ones. And if one of the precursors to that, because all of these things are coming from a similar undifferentiated hematopoietic precursor. If that precursor becomes cancerous, then what you get is like this huge wave, basically, of undifferentiated stem cells that flood this person's body. And actually, there are a couple really cool therapies out there that target the specific genetic components that cause that undifferentiated cancerous cell population to differentiate normally. And once they're differentiated, they die just like normal neutrophils. And so there's a couple cool specific things that can be targeted and treated, and the success rates are really high. But the last ditch effort is definitely to just get rid of the person's entire immune system and start over.
Yeah, I missed part of that. My cat was being annoying.
No, I know that feeling.
Matt knows about the cats being a pain in the ass.
Yeah, recently.
But when in the case of having to do the transplant, since it is an immune system from a entirely different person, there's the risk of it developing the risk of it attacking the host into which it's transplanted. Which they refer to in this paper as graft versus host disease. And so that, of course, sounds like a pretty serious problem, having an immune system attacking.
Yeah, GVHD is like all of the autoimmune disorders rolled into one and blasting you, and it's not even your own immune system, which is the really sad part. But
yeah, but it's interesting, and they talk about this a little bit in the paper. There's also this sort of, you don't want graft versus host, obviously, but you do want a little, because it's
attack the cancer.
Yeah, exactly. So in this cancer patient that has a lymphoma, their immune system has gone haywire, and so you try to ablate their immune system, but that process is never 100% efficient. So you'll get rid of 98% of the cancer, but the 2% of the cancer will eventually grow back out if you don't get rid of it somehow. So when they do these bone marrow transplantations, they're trying not to get an overwhelming response against host, but without any response, the cancer has a tendency to come back. So it's this really sort of awkward balance that people are trying to strike when they're doing this.
Yeah, and I should say, Jesse, you can feel free to cut this out later if you want to, but maybe this is just my memory fading, because I feel like at one point I understood graft versus host disease.
Yeah.
And in the last few years when I've thought about it, I've decided that it doesn't make any sense, because you're starting from bone marrow. So presumably the B cells and T cells that come out of this are all going to have to go through positive and negative selection. And you're going to have hematopoietic cells in the thymus and in the bone marrow to negatively select against cells. And you're going to have stromal cells from the recipient. So maybe I should back up and say that the reason that graft versus host disease happens is because you have essentially T cells that are recognizing the recipient cells as foreign, right? But I don't really understand how those cells make it out of thymic selection.
So I think part of the problem and part of the solution, actually, that this paper tries to address is that you've got active T cells, like fully grown T cells, that are going to come over in that bone marrow transplant. And so there is it's not just the precursors that are going to be a problem. And you're right, eventually new precursors are going to have to go through thymic selection, but you do get direct activation of fully mature T cells that have been transferred.
Okay, so you don't deplete the mature cells
from
the transplant.
They have started to do that. They've started to do depletions, and I think they mentioned that at some point during this paper. But yeah, sometimes it sort of works. The sorting process is not totally efficient, and so yeah, you do get T cell transfer.
Okay, that makes sense then.
Yep.
Yeah. All right, so this was a paper published in Blood,
Great name for a journal.
No.
Blood.
I'd like to say I've published in blood, but I would never,
Yeah, right.
probably never will.
This paper was written in blood.
I mean,
Yeah.
Or at least published there.
maybe my dissertation at closest.
Right.
And it's looking at the ability of a particular virus that people have looked at as an oncolytic virus to help with this graft versus host disease. So myxoma virus in particular, it's a rabbit virus, like the
Correct.
textbook example of attenuation, I think, in textbooks.
Is this the virus that they
Yes.
tried to infect the Australian bunnies
Yeah.
with? Okay.
Yes.
Apparently still
So
are.
it's a pox virus. So it's related to smallpox and vaccinia virus. Or yeah, I remember we got in a lot of trouble last time we talked about smallpox in our
Yeah, we did.
in our podcast. But hopefully I'll get it right this time. Yeah, so it was released in the 19. So it only infects rabbits. So they're very rarely will it infect a healthy human. And so in the 1950s, they released this virus in Australia and actually did it in France, the UK, a couple other places as well. And the idea was we're going to kill off our overgrown rabbit population by giving them this rabbit pathogen.
How'd that go?
And it went really well at first.
90%.
90% of rabbits dead. But after 3 years, all the rabbits that were left either became resistant or were just because of, like Darwin and evolution, that there's some sort of variation in the population. And so all the viruses or all the rabbits that were susceptible to the virus died and the resistant ones grew out. So after a couple of years, all the rabbits came back.
Mm.
And now they were more resistant to their original pathogen. So that was unfortunate.
Yeah.
And I
But they
think
tried building a fence across the entire continent, and that failed too. So they
should have
the
had Trump
Australians
do
are-
it.
right. The Australians are a bit of a quixotic people, I find.
I do find that, so there are all these examples of using myxoma virus as a population control mechanism, and then now they're using it as a cancer cell treatment, which is, in theory, a cancer cell population control mechanism.
Yeah.
And I do wonder
Yeah.
I did not make that connection.
Yeah, I do wonder what are like the evolutionary trade-offs in this system, and would you expect the cancer to evolve away? But maybe we'll talk about that a little bit down the line.
Yeah.
But it is something to think about when you have two biological entities. You pit them against each other, and you assume one of them's going to win.
Right.
I guess with cancer, there's another form of selection, which is the immune system of the body. Well, maybe not in this case. I don't know.
Yeah.
Yeah, well, it's interesting because we do know that cancers evolve because a lot of times cancers will evolve resistance to chemotherapy. So in the same way that bacteria can develop antibiotic resistance, well, it's not quite the same, but in a sort of analogous way to the way that bacteria acquire antibiotic resistance, and using antibiotics will select for the resistant strains, we have similar problems in cancer. And I think that, with the rise of a bunch of these different cancer treatments, like using checkpoint therapy and a bunch of other things that are trying to get the immune system ramped up, they're also going to suffer from these same problems. And maybe a combination approach.
Yeah, I'm not convinced, because especially in a case like this where you're not targeting the cancer really directly, and I know that they're saying that they're targeting the cancer directly, but I have a tendency not really to believe that entirely here.
Should we explain that real quick?
We should actually explain the paper, maybe.
Yeah.
Wait,
yeah,
no,
yeah.
I think we should just keep going off on tangents.
The last thing I said.
And we should make it like ours. We should just
that's usually
not
our
ever
bet.
actually talk about science. I think that's more fun.
So myxoma virus is normally this rabbit virus, but there were observations that this group had made before and other groups had made that showed myxoma virus is capable of replicating in cell culture in cancer cells, and then they transplanted some human tumor tissue into mice, and they believe that myxoma virus was also replicating in just the human tumors and not in the rest of the mouse. So they, even though it's normally a rabbit virus, they also there's some evidence that supports that this virus also replicates in human tumors. So that's how this becomes oncolytic. So it only replicates in a human cancer as opposed to a wild type or a normal, healthy human cell.
A normal, healthy human cancer.
Yeah. So something about the cancer makes it either more susceptible or less resistant, which are kind of the same thing, but from two different approaches to this virus.
Yeah, so there's some data they think maybe there's a couple different ways, right, that you could a virus could enter or could infect a cancer cell but not a healthy cell. Mm-hmm. And a lot of proteins are dysregulated, and you get upregulation of particular proteins on the cell surface of a cancer. And so there could be just a receptor that the virus needs to get inside of a cell is only present if that cell is now cancerous and is upregulating all these things randomly. And so now the virus can enter. There's also other mechanisms where maybe viruses enter into the cell normally. There's no problem. It can get into a cancer or a healthy cell, but only in the cancer cell where like tumor suppressors are now inhibited can it replicate and actually kill the cell. And maybe in a healthy cell, the virus is just stopped immediately after entry. So there are like some differences in biology which allow the virus to replicate only in the cancer cell. And in this case, I don't think anyone knows what it is. But we have seen examples of both for the other oncolytic viruses.
Based on the results that we'll get to, it might seem to be more of the latter because of I guess I can't go too far into it but I mean, they do see like binding and infection and replication of the T cells.
Sure. Yeah, it does seem to be that the replication is really the problem in a healthy cell rather than a cancerous cell. So we'll get into it almost immediately. Maybe that's a good segue into Figure 1, actually. So, yeah. So they really, they're basically just taking patient cells, they're pulling them out of the peripheral blood, and they're isolating T cells specifically because they're interested in T cells. And so basically what they're doing is they're taking these T cells and they're just mixing it essentially with this virus, this oncolytic virus. And so you can take healthy T cells and you can take a fluorescent version of the virus and mix the two together and just ask the question simply, does the virus stick to the outside of the T cell? And the answer is yes. So if you take a look at T cells that have been mixed with this virus that's fluorescent, the T cells are all of a sudden fluorescent even after you wash them really good. And so you can say, I would say, at least based on this FACS plot, that you can certainly get binding towards the T cell. Mm-hmm. But the real question is, in the massive number of healthy T cells that you are getting binding to, are those viruses infecting the T cells under normal circumstances, and is that infection productive? And so basically over the course of the next several panels, I don't think we have to go into the details, what you see is that if you take those T cells and the T cells are just sitting there and hanging out and not really doing much of anything, then the virus has a really hard time sort of ramping up. But if you instead stimulate those T cells, you make them really active, that's when the virus really kicks on. So it seems like the virus needs something related to the activation machinery, basically, of the T cell. And I think a couple times over the course of this, they talk about the non-specific activation of T cells. This is a more methodological thing, but if you're reading and following along with the paper, they use these beads. They're anti-CD3 or CD3 CD28 beads. And so basically what you're doing is you're taking these T cells, and T cells require two signals. They require, first of all, that their T cell receptors, so this is their main receptor, and it's what gives the T cell specificity, that receptor signals through a complex that we call CD3. So basically on these beads is an antibody that tickles that CD3 receptor and gives signal 1. And signal 2, or the co-receptor, is this CD28 molecule that they're using. So basically what happens is when you get T cells on these beads, both signal 1 and signal 2 are presented to the T cell. The T cell recognizes that as an activation signal, and so it starts proliferating like crazy. And in the case of the infected T cells, when it starts proliferating, or later we'll find out not proliferating, really, when that machinery kicks on, you start to get viral production.
Yeah, I should say, or I wanted to interrupt earlier, but it's not really that these antibodies tickle the T cell receptor. It's more like socking them in the face.
Yeah, it's not even
such
close.
Violent imagery today, Kevin.
I'm sorry.
I think you've been listening to Trump too much.
Yeah, maybe.
Yeah.
They're making T cells great again. No, so these antibody cross-linkers are basically smashing the T cell with activation signal, right? Like this is not going to be a physiological level of T cell activation, and you're going to hit every single T cell that has a T cell. I mean, it doesn't matter what the specificity of that T cell receptor is, you're going to be activating it.
Right.
Yeah.
Is there something about the CD3 T cells that distinguishes them for this study?
No, actually, so CD3 is just a marker of all T cells, because the T cell receptor itself, the part that actually binds to the ligand peptide MHC, it doesn't actually have an intracellular, I mean, it's got a tiny little tail, intracellular tail, but it can't actually signal on its own. So CD3, there's actually several different proteins that make up CD3, but all of those are required in order to propagate the signal from the T cell receptor. So CD3 is a non-specific T cell stain, basically. So all T cells are going to be marked by that.
Okay. So this is just signaling. This is just stimulating and testing all of them from the donor. Correct?
Right. So if you have T cells in the soup and you hit with CD3, you're going to hit every T cell. But I think do they sort
They
on
do
the type
break
of
it.
T cell before then?
They break it down into CD4 and CD8 in 1D and 1E and,
Right.
a couple, but they never really get more specific than that. So there are these two broad classes of T cells, at least the best known T cells. There's these two broad classes of CD4 and CD8 positive. So your CD8 positive are going to be your cytotoxic T cells that are going to look for infected cells and then kill them, where your CD4s tend to be more of the helper variety. They help other immune cells do their jobs effectively.
They also mention Tregs.
Mm-hmm.
Are those
Yes.
those two?
So those will be CD4 positive, but they kind of go in the other direction. So we think normally of T cells as these pro-inflammatory T cells. Even CD4s will tend to help other immune cells in their function, but it's usually a pro-inflammatory function. Tregs are a little bit different in that Tregs do, basically just the opposite. They will find and shut down the function of other CD4s. So if most CD4s are sort of this positive pro-inflammatory signal, your T regulatory cells are going to be your negative regulators and your anti-inflammatory signals.
Yeah. So what they saw with the virus was it was when the cells were stimulated, it was infecting them more often and causing some of them to proliferate, but not the Tregs.
Right, and I don't know, how much do Tregs, when they get stimulated, do they blast to the same level as other CD4 T cells?
I still think that's so weird.
What's that?
Blast means divide.
Yeah.
That's not violent imagery. That's not the influence of Trump. That's actually a technical immunological term. We say
Right.
cells blast.
Yes.
So
I don't think that somebody will yell at me because I don't know nearly enough about Tregs. We should get Dimitri in here, but I don't think that they divide with the same rigor as other cell types.
okay. Well, Stupid cat.
Yep.
So-
Can everyone hear my cat? That's my cat.
I was wondering who that was.
Yep. That's mine. He's trying to
Feel
get out
real
the
bad
door.
that all three of you have cats, and I don't. And I really love cats, and I miss my cat.
Oh, you can have mine.
No, my wife's allergic,
Oh,
unfortunately.
well, definitely don't want that one then. All right. So, general question to everybody involved in this. What are your thoughts on including full and partial responders in Figure 2 as separate groups?
To clarify, those are the ones that proliferate, I think.
Yeah, so we go from Figure 1, which basically says that the virus likes to hit T cells, and when those T cells get active, you get a lot of viral production. Mm-hmm. And that's great. And then we move into Figure 2, and basically what they're saying is that of the T cells that get infected, when you try to stimulate them, they don't blast, actually, to recall the term. They don't blast the way that T cells normally do. They don't divide the same way. But when they're looking at how well these T cells are dividing, what they're finding is that sometimes in certain patients, the T cells definitely don't divide at all when infected with this virus. And then other times, they sort of divide. And so there's kind of this partial phenotype. And in order to, I think, run the statistics, they basically separate the full and partial responders into two different groups and then consider them statistically independent of each other. And I have to say that really bothered me.
Well, I think that's a perfectly reasonable thing to do as long as you can account for that in your statistical model, right? Like when you are doing the tests, if you do it correctly, you can basically say that you subdivided your population, and then you have to do things a little more stringently in order to consider them significant. But that's not totally unreasonable if they have enough power within these different subsets. And I think it's worth trying to divide them in order to figure out what is making some respond and some not respond.
Right. So my follow-up to that was, did anyone see how they subsetted those groups or anything about any of the statistics in this paper in its entirety?
No.
Yeah, no.
Nope.
Okay, because my follow-up to that is I don't believe the statistics in this paper. And partially, I believe that the effect that they're seeing generally over the course of this paper is real. They're certainly seeing some differences, but there are some major red flags, to me at least, in how some of these numbers were crunched. There are some panels in this paper that some of the bars just don't have error bars. They have p-values, but there are no error bars associated with them. There are some where the error bars are similarly overlapping in two different conditions, and one has a p-value of 0.009 and one has a p-value of 0.25, and none of it makes sense to me, really. So we don't have to dwell on that, but I would call into question how some of the statistics were done here.
It's kind of sketchy.
Yeah, but overlapping error bars are not equivalent to p-value. So.
Agreed. No, totally agree with that. But, I mean, so if you look at Figure 4B...
Yeah, we're only skipping ahead a little bit.
Yeah, I know.
No, I saw that.
Yeah.
I did.
That's concerning to me.
Yeah, but, I mean, it depends on the number of observations and et cetera, et cetera.
But I have no information to support how many observations they had.
That's
they
fair.
never told me, and they never told me what tests they're running. So, anyway, we don't have to, like I said, we don't have to dwell on the statistics. It's boring to do that.
So where the hell are we?
I think we're on Figure 2.
Yeah, we're in the so many fax plots.
Yep.
So many plots.
Yeah, this is like kryptonite for Kate.
I just see them, I'm like, oh, sum it up for me.
Yeah. Well, I think the summation really is that if you've got a T cell and it's infected with this virus and you try to stimulate it, so we saw in the previous figure that a T cell infected with this virus that's stimulated will start producing more virus, and that's how the virus sort of knows when to turn on. But what we see in figure 2 is those T cells really aren't dividing at all when they get turned on. They're just sort of, they're trying to divide maybe, and virus is being produced, but they're not dividing like they normally would.
Is that a kind of result that you would want to see when transplanting?
Well, it's certainly weird because typically the first thing that happens when lymphocytes get activated is that they start to blast, right? They start to divide over and over again. And that's like a pretty typical assay to check for activation is if the cells are dividing. So if the cells aren't dividing, then that suggests something kind of weird is happening.
Yeah.
I would say from a clinical perspective, we were talking before about the possibility that adult T cells would be transferred from the donor to an immune-compromised recipient, and some of those, mature T cells are going to be a problem potentially in graft versus host disease. This might actually suggest that it could be beneficial to not, to introduce this virus into the donor cells, because if it is truly limiting mature T cells' capability to divide, maybe you'll limit their potential to do harm in the recipient. But that, I think, is pure speculation.
That's what I was thinking.
Yeah.
Yeah. I'm not very convinced on their infection, to be honest, though. I mean, oh, go ahead.
No, as far as the quality of their infection?
So they have they bomb these cells with virus, like an MOI of 10. You expect every single cell to be infected if there's going to be a productive infection, depending on how you titer the virus and whatnot.
Sure.
But they don't even graph their their replication. You don't see anything beyond one cycle of replication, so you don't know how much is really coming out. Mm-hmm. And then they show a number of cells infected, but they give it to you as a linear. They try to show it's growing, but it's on a linear scale. So these aren't actually growing exponentially. So I'm not really sure that these are infecting, but I'm not really sure these are infecting the T cells. But on top of that, you really don't want them to be strongly infecting your T cells.
Right.
So I don't think it's a problem, but I was just very confused why they kept saying these cells are infected. I'm like, really?
Well, we said that we don't know. We don't know what is making these cells permissive when, normally human cells are not permissive to infection, right? So maybe it's not completely permissive.
But these aren't the T cells. The T cells aren't supposed to be cancerous. These are just supposed to be wild-type cells. Right. Yeah. So these cells theoretically aren't permissive. At a certain point, there are plenty of examples where if you put enough virus on cells, something's going to get through. Sure. So especially when you start having effects from super infection, so you're going to have multiple viruses entering a cell, lots of weird things can happen. But the infection isn't going to actually, I don't think an infection is established here.
Yeah.
You're probably not going to get multiple rounds of replication. You probably blast these cells with virus. Some of the virus gets in, and it does something weird to these T cells, but it's not really an infection that's occurring. You're not getting productive infection. It still looks like it's probably an abortive infection. This is going to be something that a virologist is going to care about, but
sure.
For the purposes of this and looking at the effect of the immunology, maybe you don't care as much about. So,
yeah, I actually want to bounce off real quick the MOI that you just mentioned. So they mentioned that they've got an MOI of 10 throughout this paper. Basically, every experiment they do is between 1 and 10, but most of them are 10. And in the first paragraph of their results section, they say that the lower limit of sensitivity of the binding assay with the venous tagged virus is about 500 virus particles per cell.
Yes.
Mm-hmm. And I don't understand how you could get 500 virus particles per cell if you've got an MOI of 10. Is that just me being dumb?
No. So it has a lot to do with, theoretically, when you say MOI, so MOI means multiplicity of infection, and it usually means how many virus particles per target cell are there. So you would, so it's a ratio. Theoretically, it's a ratio of particles to cell. But in reality, very few people actually have a absolute particle count of their viruses that they're using. So to get a ballpark of how much virus you have, you usually plaque your virus. And what that means is you make serial dilutions, you infect a cell monolayer, and then you count how many infected cells there were. So one virus will infect a cell, and it will make a small clearing in that monolayer. And so what that gives you is a measure of infectious virus that you have in your viral prep. And so a lot of people will say like their MOI is like 5 PFUs, so like plaque forming units. But you might have a lot more virus actually in your prep.
Okay, so I guess here's my question associated with that, and I agree with everything you just said. First of all, would you expect that number would be 1 in 50 particles might be infectious? Is that a reasonable number?
It really depends. So something like HIV, you're almost at like 95% infectious.
Right.
But then other viruses, like some other RNA viruses, they have a lot more defective particles. So particles that won't give you a productive infection. Maybe they'll enter, but then they'll just stop the infection.
Right. I guess the reason that I ask is because they do this FACS plot, right? So they're looking at cells that binds this fluorescent virus, and they say that it takes 500 viral particles to get over the limit of detection, right? And the limit of detection, from the lowest limit of detection to the highest positive cell in that assay is 3.5, 4 logs. Right? So the question is, with an MOI of 10, do you think you can load 500,000 viral particles onto a cell?
So I'm
putting you on the spot, and I know this is not your system. So, the numbers didn't make sense to me. I guess that's why
I'm not sure. My guess is that there's a lot of defective particles there, or just maybe I don't know how they did their viral preps.
Yeah.
I will say though that with the MOI, just because it's a ratio of 10 viruses to one cell, you would not expect to have 10 viruses entering one cell. So the actual, what I would say is you would expect less viruses than that per cell. So like an MOI of 10, but then you're seeing 500,000 cells per, or 500,000 viruses per cell. I would say that sounds like you are greatly underestimating your particle number with your MOI.
Yeah, that was my thought too.
Because usually like an MOI of 10, you would expect like 3 to 4 viruses per cell, which is a ton.
Right.
But if you were looking at infectious units, then you're looking at what am I trying to say? If you calculate your MOI by infectious virus, you're going to be eliminating a lot of viral particles, just so many. So my guess is that they probably have some strange way of titering their virus.
Yeah, my concern, I guess, is that they've got free Venus. That's the fluorescent protein that they're using. Mm-hmm. That they've got free Venus in whatever they were dropping onto their cells, and what they're looking is not actually viral attachment. It's nonspecific binding to some free protein.
Oh, I would definitely have that. I definitely think that's probably an issue that's going on.
Yeah. Anyway, yeah.
Oh, yes.
So, regardless, I think that certainly they've got virus in the solution as well, so I don't think it's too much of an issue, but it's a bit concerning.
Okay. Are we on figure 3 now?
Not sure. So we can be. Right. So you can imagine that if these T cells, we mentioned that T cells that are infected with this virus that get stimulated, first of all, virus starts replicating, and then somehow the T cells are inhibited from replicating. And so the question is, what's happening to those T cells? And one might imagine if T cells are trying to replicate but can't, they're probably freaking out a little bit. And so you might expect that some would die. Mm-hmm. And so basically that's exactly what they're looking for. They're looking for these viral infected T cells. They stimulate them and they ask the question, how many of them are dying? And the answer is, it depends on the person. We have this full responder and partial responder effect, but regardless, a pretty significant number of these T cells, when you're activating them, are dying pretty rapidly within 72 to 96 hours.
Yeah, pretty consistent, right?
Yep.
And only when stimulated. Yeah.
Mm-hmm.
Right.
So, good.
Similarly, I think just kind of moving right on to their function, right? If these T cells are dying and they're not proliferating well, you'd probably also expect that they're not going to be producing a lot of the cytokines that they would normally produce. So when T cells get activated, they tend to produce things like IL-2, which helps themselves divide, but it also helps the T cells around them divide sometimes. And you get, what else are they looking at here? They're looking at...
We have some IL-10, IL-4.
Yep, some interferon gamma. Yeah, so basically just things that T cells produce normally. And as you might expect from a group of dying T cells, they're just not going to be able to express those cytokines as well.
Yeah, which I would also say gives more evidence to the fact that the virus is not going to productively infect these cells. Because if these viruses need activated T cells to infect the cell, and when you infect the cell, they then turn off activation cytokines, you're going to start, like a quickly decaying cycle of infection, or cells that are capable of becoming infected.
Right.
Yeah. Okay.
Did anyone understand Figure 5?
No.
Figure 5. Let's rewind this.
So we've got some mixed lymphocyte reactions going on.
5 and 6.
Yeah.
You know what? I don't know what MLR is.
It's a mixed lymphocyte reaction.
Oh, okay.
And so usually what you can do is you just hematopoietic cells grow like dirty preparations of hematopoietic cells. When you mix them together and they're from different donors, have a tendency to activate each other, and you get this big explosion of immune function in the dish.
Okay.
And so I think what they're looking at is just how this virus influences the ability to participate in a mixed lymphocyte reaction. But to be perfectly honest with you, I didn't look at these figures all that closely. I apologize for that.
I think one of the things they're looking at is also, so the first one is, if you have the mixed lymphocyte reaction, when these cells are activated, can they now sustain an infection with this virus? And the answer is yes. So you have like higher levels of viral infection when you have the mixed lymphocyte. But then the second one is looking at proliferation. So you get a lot of right? So you're getting like a lot of proliferation of PBMCs or the cord blood in these mixed lymphocyte reactions, but you don't if you just have the responder stimulator cells. I mean, it's a pretty simple hypothesis. Like, the virus requires activation for infection, and when you have lots of activation, they become permissive for infection.
This is getting a lot longer than I usually go. I think we could skip to Figure 7.
Absolutely.
Right, so here they were looking at actually how the T cells behave with the multiple myeloma cells transplanted or taken from human donors, with or without the virus and with or without the stimulation of the T cells. So They say that previously in the mice studies, they had seen positive results when treating with the virus. So this means the T cells were less likely to cause problems for the host, but not less likely to cause problems for the cancer, which is exactly what you want.
Mm-hmm.
And so in this case, they tried doing that in cell culture.
Yeah, I mean, it's sort of the perfect system, right? If you're looking to limit the donor's T cells' ability to kill off your recipient, and you're looking to potentially infect myeloma cells and kill those directly, this virus seems to do both of those things at the same time, at least in the system they've worked out here. So they've got this nice cartoon showing that the virus can be transferred between the T cells and the cancer cells, which is great if you're the recipient. So I don't know what's not to like.
Yeah, it's not only that the virus is killing the cancer, it's actually stimulating the donor cells to kill it better also.
Yeah. And it seems to be limiting the donor's T cells' ability to kill the recipient. Mm-hmm. So, yeah.
Yeah.
Not bad.
Yeah, it's a cute system.
Yep.
Seems to have good potential.
Yep. Kevin apologizes, by the way. I don't know if you saw that email.
No.
He lost power and internet. So he's going to try to hook up to the YMCA. So we'll see if that works out for him. I don't know. So what did you guys think? Do you buy it?
Yeah. I mean, I think it's an interesting system. I like different therapy approaches. I think those are really interesting. Just in general, I'd rather read something about a crazy new virus than I would about listening to any more ad vectors. Very sick of ad vectors.
Yeah.
Adenovirus vectors. But I think one of the interesting things about this virus is that it doesn't look like it should cause a disease in humans, which is probably one of your number one safety concerns when you're trying to think of a viral therapy, is to making sure that you take out all of the harmful parts. But if you have this virus that is just so weakly infectious in a healthy human, I think that's a really great start. I would want to know a lot more about how this virus works in a human, and I think that this is a really good start. But let's look at like the T cells, and especially if the T cells are going to have something to do with transferring this virus to the site of the cancer. I think that this is a great start. Does it infect them? What does it do to them? Are you going to cause some secondary problems by having this virus? So,
They did say that they tried with immunocompromised mice and didn't see significant problems. That seems good for safety.
Yeah, it's a start.
Mice anyway.
Yeah, it's a start.
Yeah.
And you'd probably need a paper. You definitely need a mechanistic paper here showing what's actually going on. I feel like the major concern that I have is, there's some paper concerns, but that's fine. I think that the observations are probably true. I think these donor T cells are certainly affected, and you seem to get oncolytic function. I do wonder what FDA approval on something like this would actually look like.
Yeah.
Because you're claiming that this virus is going to do like 3 separate things. One of them is target the cancer, and targeting cancer with viral vectors, that's gone through a couple times. But what concerns me is that you've basically got huge amounts of binding of this virus to the cells that are going to be responsible for sort of saving the recipient. And so that sort of modification of the donor bone marrow, I imagine that would be pretty hard to push through the FDA.
It's pretty much a treatment for the donor cells on the way to the recipient, if I understand correctly.
Yeah, exactly. So you're going to pull the donor cells, and then you're going to mix them with this virus, and then you're going to, basically mix it all together and then pump it all into the recipient. And so you're pumping probably live virus. You're certainly pumping modified cells, if you believe this paper. And so the question is, how easy is it to get past something like the FDA, the fact that you're modifying the bone marrow before it even goes into the recipient?
Yeah. I'm wondering how, since it is like a one-time treatment, how long-lasting is this effect? I don't know how long-lasting it needs to be. I guess if it's just preventing the T cells that get transferred that one time,
yeah, then
it could be useful.
Yeah, it seems like it could be. And, if it helps bump that small bit of needed graft versus host so that you kill a couple more cancer cells along the way and you're a little more, it could work.
Yeah.
It could work.
At least up the chances of it being effective.
Sure.
Yeah, sounds pretty interesting.
Yeah, good stuff.
I thought it was interesting that some of the funding came from the Gatorade Trust.
Like sports drink?
I think so, unless there's another kind of Gatorade.
Yeah. I did see that, but I don't know quite what that means.
Yeah.
But
well, thanks for joining me.
Sure, thanks for having us.
Thanks for having us.
Pretty fun and more knowledgeable than I would have been on my own. Definitely.
Yeah.
It's cool to talk about viruses in this way as potentially helpful.
Yeah, I like the idea of making biology work for us.
Yeah.
I feel like we're on the cusp of engineering some really cool things that help us out.
Yep. So we'll see if this goes somewhere in the future.
Hope so.
Okay.
Cool.
Guess that's good.
Cool.
Awesome.